The next study is from Dr. Jeffrey Friedman at Rockefeller, looking at the cellular and molecular basis for obesity and PWS. And the rationale here is that we know that the hypothalamus is likely important in regulating appetite in PWS, but we don't know the specific cells within the hypothalamus that are kind of driving the deficits in PWS. And there are new technologies that the Friedman lab is using to identify cell type specific changes in how these cells function. So Dr. Friedman, who is, really, again, another giant in the field of obesity, his group identified leptin, one of the first regulators of appetite known. He's going to use this multiplex single nuclei RNA sequencing, again, a relatively new technique, and apply it to identifying the specific neurons that cause hyperphagia in PWS, and use that information to screen for drugs that might correct these deficits. So this is a newly developed and powerful tool. Data from this study should be useful to many researchers in understanding the differences at a cellular level between the hypothalamus in PWS and the hypothalamus in typical individuals, or mice in this case. A very experienced researcher in the obesity field, so really has an understanding of what drugs are out there and might be applicable to PWS. And this group has had success in applying this approach to other obesity models. So the long-term goal here is to identify the underlying cause of hyperphagia and then validate new drug targets that might be useful in PWS.